Now, I would like to look towards the future of snake breeding because I see this happening in some of the other I guess you could say animal breeding projects that we see in dogs and cats and stuff like that, and in science in general. I mean, just going to my end of the world, which is bodybuilding, and performance enhancement. Gene editing of humans seems to be what's going to be the next frontier in creating the perfect beast, so to speak. We'll take out the stuff we don't want. We'll add in the stuff we do want. People who work out but athletes want more muscle and so they have more power and explosiveness and competition. Bodybuilders want to be bigger. And so we have these gene editing techniques now known as Crispr, where you can cut genes out using viral vectors and insert new genes into the animals or humans, whatever you're experimenting on. I was reading an article about how they're going to be, basically gene editing dogs for different colors and patterns and eye colors and stuff like that using these techniques. And they might actually already be doing it from what I understand. But do you see application, towards the reptile breeding industry? You think that'll make its way over there as well? And explain what Crispr is, just for people who don't understand.
All right. So Crispr is it's a gene editing technique using basically co-opting what is a bacterial immune system response to bacterial viruses. So the viruses that attack bacteria, they've created this immune system response that allows them to chop up those viruses when they come in. And the technology has been co-opted by scientists to be able to make very, very fine, detailed changes to DNA. It's almost like autocorrect in DNA. Now, I say almost, actually, that's probably a very good analogy. If you've ever used autocorrect on your iPhone when you're sitting there typing away, sometimes it's really good. But sometimes I am not talking about the worst ducking luck on the planet.
Right. Sometimes really off.
Autocorrect makes a correction that is horribly wrong. And we're still there with CRISPR too. Sometimes you can make the correction the right way, but we haven't got the resolution 100% to be saying that there will never be any negative side effects with it.
It's scary because
there's a real big moratorium on using this in human embryos right now, because it's not just the whole scary idea of, now you're going along the lines of eugenics and, taking what Hitler wanted to do to the next level of stupid. But you're also looking at the real problem of, even if you're trying to make a beneficial change, like if you're trying to correct hemophilia or Tay-Sachs or Huntington's disease, if it goes off target, you've potentially created another problem while trying to fix that first problem.
You could create an abomination, really, is what you can
do. Right, and that's, in the science field, that's what we're more concerned about. And this is one of the things with scientists, is we really like to try and look at this through these kind of things through rose-colored glasses. I know all the scientists want to do this for the most pure reasons, curing genetic diseases and stuff like that. But there's always somebody out there that wants to do something wrong, and the scientists don't think about that because they really are trying to go more for this altruistic way. For the mad scientists out there, because we've got to have our supervillains for the superhero to go after. But there's that moratorium against human experimentation. It's less so with animals, because even though everybody likes to say that animals shouldn't be experimented on, animals are a lot more palatable to experiment on, because if you mess up in something like a mouse or a guinea pig or a zebrafish, it's not the end of the world, and people aren't going to feel terrible if you then take that mouse or guinea pig or zebrafish and euthanize it and put it down. And we have been gene editing animals for a long time, even before CRISPR. It wasn't the cleanest way of doing it, but those glowfish that you see in aquariums.
I was going to bring those up. Yeah, those are from that, right?
That's a jellyfish gene that was inserted into them, and that was originally done in science labs to understand cell differentiation and be able to track how cells divide and split over time.
Awesome.
And then somebody who was working in a lab, talked about it with their friends, and somebody was like, that would be really awesome in a fish tank. And then at the end of an experiment, rather than put all those fish down, they convinced the lab, hey, I'd like to take these fish.
And they made a lot of money, I'm sure, from that.
Multiple times because, axolotls are, a real big pet in the industry.
Sure.
There are green glowing axolotls.
Are they really? Wow.
The exact same thing. It's the jellyfish gene that's been inserted into them so that they glow. For the same reason, it was done to be able to track, cell differentiation and cell proliferation, cell regeneration. And then it got out of the lab and into the hobby, and now you can buy a glowing green axolotl.
Now, what's to stop you and I from starting, Glow Ink and making a glowing ball python? I mean, is that something that could be done with CRISPR?
It's something that could be done with CRISPR. It could be done with more classical genetics. It would be a lot harder with classical genetics. It's a little easier with CRISPR. It's still not the easiest, especially with things like reptiles and stuff, because they're laying eggs. And so, getting an ovum out and changing it and then getting it back into the animal,
you're right,
is more difficult. It's especially difficult in the more classic genomic sense where, with when they did it with mice, they could extract an embryo with a mouse and put the changes into the embryo and then insert that embryo basically through IFV type techniques into another mouse mother. We can't do IFV in snakes and lizards.
Why can't we just put it right into the follicles that are developing?
And that's what they're doing now.
Brilliant idea.
There was a group.
Yeah.
If anybody's interested, track down this paper.
What's it? Read it to us. What is it?
It's CRISPR-Cas9 gene editing in lizards through microinjection of unfertilized oocytes.
Oh my God! And it works, huh?
And basically, what they did was they used CRISPR. They make a little microsurgery incision into the lizard.
Right.
They insert it into the ovum.
Crazy.
The CRISPR change happens, and then you get an albino anole.
Wow. Think about take an animal that has no albino right now.
Right.
For instance, I don't know, an alligator.
We have albino alligators.
Not alligator. I'm saying an alligator. What are those things called? The ones
Alligator
that Tom Crutchfield that Tom Crutchfield has. Oh, those. Crocodile. Excuse me, a crocodile.
Monitors?
Yeah, crocodile monitor. Thank you.
Yeah.
I don't know why I had a brain fart there. Imagine making an albino one of those.
Right.
What's the price tag on that?
Exactly. And you can do it with all kinds of things. Now, again, caveat with most of the morphs that we have in the hobby, we don't actually know the gene that causes the morphotype. Albino is really well known because we've characterized albino in pretty much every animal out there that it's occurred in. It's the tyrosinase gene, and we know that very well, which is why they used it in that paper.
Interesting.
We know that we can knock out the tyrosinase gene. We know that we can make an albino. But, there are a lot of lizard species out there, a lot of really popular lizard species out there that don't have albinos. What about an albino bearded dragon? What an albino crested gecko? People would probably lose their minds over something like that.
Absolutely.
And
I can't believe it hasn't happened already if the technology is there.
The technology is there, but the technology is there inside of the lab because,
You got a lab, though, Travis.
I don't have free reign in my lab to do these kind of things.
Right.
I can't bring a lizard into the lab and be like, hey, I'm going to perform microsurgery on this lizard. And, most hobbyists don't really have the ability.
Your life right there,
Yeah, most people don't have the ability to do microsurgery on lizards in their home basement or something.
Right.
But there are probably small labs out there or labs out there that could work in collaboration with, larger breeders where it could be possible. I don't know who those people would be, but I would guess somebody out there might have that ability. So
I can't even imagine that it isn't already happening, or now that we mentioned it, people will start,
yeah, people are going to start calling labs at random and be like, do you want CRISPR? Hey, I've got a lizard, can you make it albino?
Right.
Don't just call people randomly, guys. It doesn't work that way.
And here's the question I think probably a lot of people have, I know the answer, but once you gene edit and you produce these albinos, they will then reproduce as albinos?
Yes, well, they should. The gene editing done by CRISPR is done at the base level of the egg. So then once the egg develops and everything, the ovum develops, the fertilized embryo develops, it's in all of the cells in that animal. So then the gametes that it produces should carry that mutation on.
Wow. I really would like to see a glowing snake, I think, rather than an albino. If I'm going to do CRISPR, let's make the thing glow,
Yeah, but bear in mind, like the glowing fish and the glowing lizards, they only really glow well under UV light.
Oh,
and
interesting.
Most of us tend to keep our snakes in tubs where we don't have a UV light. Now, if you've got a great big,
lizards, though, they have UV light.
Or if you've got a great big 4 foot or 6 foot or 8 foot PVC cage, slap an extra light bar in there that's UV, and then have it turn on at night. And at night, here comes your glowing green ball python.
Well, yeah. Well, I think that's
green anaconda.
That would be, I've been an aquarium guy for years. I always had fish tanks my whole life, and I loved them. And the new thing is setting up these almost like aquatic, garden, so to speak, and they have the nano versions
with no fish, but all the plants and stuff.
Yeah, little bits of fish in there because just the decoration. So, and now I even see in the reptile hobby, I see Brian Barczyk has the whole reptarium, and people starting to create these naturalistic enclosures for their animals just because to display them. Maybe not all your animals, but a couple of them, so to speak. Imagine having, like you said, a UV bar there, and then and these animals are just lighting up like glowing. Crazy.
Get a diurnal animal that does it. And again, there are people out there that hate me for making these suggestions, but like day geckos or chameleons, which are out during the day and have that UVB bulb on with the regular bulb, and you'll have something that's like glowing like a neon sign while sitting there. Yeah. In its little, really lushly planted vivarium, or again, I'm gonna create hate for myself and realize, guys, when I say this, it's still probably a long way away, but do it to dart frogs. Can you imagine it?
Oh my God!
Bright blue, glowing,
unbelievable, right?
Azorius hopping around. I mean, that would be pretty unbelievable. And I know that I'm going to get so much hate mail because I said that. But
you know what? Think about it.
Think about.
There's only so many genes, in the reptile. Eventually, we're going to get bored. There's not going to be any more combinations to make. We're going to have to go to someplace else. And I mean, a glowing, snake, would certainly, fit that, that need for what's the next thing we want in our animals. How about a glowing scaleless, ball python?
And there would be people that would hate it, both because it's glowing and because it's scaleless.
Of course. Well, everyone hates it. People resist change, all the time. I mean, people like to have everything figured out and pigeonholed, and it makes them feel very comfortable when they wake up in the morning, like they have a good grasp on everything. And then all of a sudden someone creates something new that never existed before, and they're like, it throws a whole monkey wrench in their whole life, because now they got to get that. First they have to resist it, and then after they resist it, they get it, and then they're on that bandwagon as well. So
and that's the thing about the hobby that I don't understand is, if you don't like one specific thing in the hobby, then just don't work with it.
Right, exactly.
People hate on the scaleless project. Scaleless isn't for me. I'm not going to have scaleless in my collection. It's just not something that appeals to me.
Right. But
that's the extent of it for it goes for me. I'm not going to work with scaleless. I'm not going to rag on people who want to work with scaleless. Sure. If you like scaleless, work with scaleless. The fur over the spider ball python. I mean, if you don't like spiders, just don't work with them.
Don't work with them, right? Exactly.
Don't say that somebody else who's working with spiders is a terrible keeper and terrible and only in it for the money. And just they like the spider. They want to work with the spider. Let them work with the spider for crying out loud. I don't like English bulldogs. So I don't have an English bulldog.
Right.
I don't hate on everybody else who has an English bulldog.
Right. Would it be? You think it'd be possible to take, a retic? I know the big thing now with retics is Garrett Hartle's a big proponent of this. They're trying to get these super dwarf, retics. Imagine, can you just knocking out the growth hormone gene or something like that, or at least reducing it, so that the animals just don't grow that big? I mean, you think that could be something where we can create? Because I know in the dog hobby, teacups. I have a teacup Yorkie and a teacup Maltese, and they're little tiny dogs. They don't even need to be walked. They go on little pee-pee pads, and they're so cute, and they stay small. You think that the miniaturization of pets would be something that CRISPR would be able to accomplish?
In theory, yes. In practice, I think it's easier to do it the way Garrett's doing it.
It takes a lot longer, though. That way,
more practically been done. Find smaller animals and breed them together. And the reason I say that is because these dwarfism traits, these pygmeism traits, it's not a single gene mutation most of the time. Other than like classic dwarfism in humans and stuff, but that classic dwarfism in humans comes with other side effects. It's not like boom, you're suddenly smaller. There's a lot of other physiological side effects. But when you have animals that through selective pressure, naturally or selective breeding over time, are smaller and smaller and smaller, if done properly, you don't have a lot of those secondary problematic defects. Now, when you take it too far, you can. Chihuahuas have, their heads don't fully form over, and they're like a newborn baby. They've got a soft spot even as adults. They tend to get the pushed in jaw and stuff, which can cause problems with breathing and stuff. But if done properly, I do think you can get smaller stature animals.
Gotcha.
And like I said, Garrett, I think, is doing it probably, the better way, because he's taking animals, he's taking small females and he's breeding them. And obviously a small female can only carry so many eggs. And so she's not going to push out 50 eggs that are too big for her. Her eggs are going to be properly sized for her body. The babies that she generates are going to be on the smaller side because of that. And then you just keep selecting for that smaller and smaller and smaller. Now, we do have to worry about inbreeding depression when we do those kind of things. So that's where Garrett, I know that he tries to do outcrossing and then back in crossing and outcrossing and then back in crossing to keep, the genetics of the whole project a lot more diverse. So it's not just constantly mother to son, mother to son, mother to son type of thing, which can create a problem.
Yeah. No, it's interesting that, people always looking to speed stuff up, though. But, they've done it in dogs. The size of dogs have gone down and down and down. And that's just from, like you said, selective, breeding. But look, you know what? I think the whole CRISPR is really, even though it's scary in one sense, it really is the future of a lot of, stuff that we're going to see, whether it be in humans or in animals, but certainly in animal breeding. I definitely see there being applications. Do you see it being so easy that, like, you might just give your animal, like the mother of the clutch, so to speak, just a shot at some point, and they'll just edit the entire genetic genome of this animal before it even reproduces? You think it'll get to that point maybe 20, 30 years down the road?
It might be a little bit more complicated than that, but it would not surprise me if it does at some point become basically a home hobby level type of thing. And, it sounds like science fiction. I get that. But in a matter of, as a matter of perspective, when you and I were growing up, think about what computers were like.
Oh my God, yeah. They didn't exist when I was growing up.
Right. This is a more powerful computer than anything we had growing up. And
you're right.
This fits in our pocket. And the time that we went from even, if we had a desktop computer with this much power, the computer that sent men to the moon in the 70s,
scary,
had less computing power than this.
That's really scary, I know, when you think about it.
If we have this much computing power, now it's the same type of thing. What we were capable of genetically 10, 20, 30 years ago, 30 years ago, nobody would have thought, we would have been able to do anything like CRISPR. So what in 10, 20, 30 years are we going to be able to do with what we see now?
We can't even conceptualize it, probably. Yeah.
Right. It's really hard to conceptualize because it's moving really fast, and it's because it's in combination with, you know, computing power and things like this, but also, you know, microscopy and molecular manipulation. It gets so much better and so much easier. So yeah, in 30 years time, it wouldn't be a surprise to me if maybe not as easy as just stick a needle in type of thing, but it wouldn't surprise me if we have a much easier ability to do gene editing on a sort of a hobby pet level type of basis. Now, that also doesn't account for what kind of restrictions are going to be put on it, because, you know, some of these things, you know, they can be misapplied. If you can change your dog, let's say that somebody isn't going to be like, well, if I can give my dog a shot and it makes my dog have different puppies,
I'll give my kid a shot.
What if I want to give my wife a shot when we get pregnant to try and make my baby better? You know,
right.
So there will probably be some type of regulation on it, simply because we don't want it going just rampant with people doing stupid things with it.
Stephen Hawking, in his last book, you know, before he died, said that he sees that there's going to be a race of superhuman, you know, people that will have access to these technologies, but won't allow the regular public to have access to them. They'll be, you know, basically enhancing themselves. And
the haves and the have-nots. And, you know, and that's on a broader sense. Again, you know, we see that, you know, it used to be that only the really rich could afford things like this. It used to be only the really rich could afford, you know, a laptop computer. Only the really rich could afford a desktop computer initially. It starts there, but, you know, this, again, editing humans, that definitely then becomes once you start editing humans and you give one group of people the ability to enhance themselves at the expense of others simply because you can outprice the others, then, you know, that's different than the stratification of, well, one day these will get cheap enough where everybody can get them. It's harder to justify that with people. Yeah. So
do you think they could ever,
Hawking was getting at there, but
do you think you could ever have a snake grow arms? I mean, do you think can it get that crazy? You know, limbs and stuff like that?
It could be difficult to get them to grow arms. Now, you know, with pythons and boas and stuff that already have the spurs, which are basically their back legs.
Sure.
I do see how we might be able to tweak the gene there to turn it back on, because it's mostly off, to turn it back on so that you get snakes with back legs. Whether they're functional, a whole other story. But if somebody wants to get really crazy and have what's basically a T-Rex of a snake running around on back legs with no arms, flopping around, I won't stop them. I'll laugh at them because that would be a horrible thing to do. But